introductions. Oh, sorry, I will mention about earthquakes. So is it okay for everyone? Okay.
Yes, we can clearly see.
So I'm opening the today's session. So hi, welcome to FLICNA podcast session number 3. Today's topic is the association between autism spectrum disorders and genetic problems. And we have a very special expert to talk about this from Germany, Dr. Christian Schaaf, who currently works in Heidelberg University Institute of Human Genetics. I will give more details about him in a bit. As many of you may already know, 2 big earthquakes happened back-to-back in Turkey a while ago, affecting both Turkey and Syria. And I want to thank FLICNA community for their support during this time, and all international collaborators who helped Turkey during these very traumatic times. As we've just begun to take it under control, another 6.4 magnitude earthquake happened just a few hours ago today. My sister is a thoracic surgeon in the area, so I wish her and everyone their safety. Seems like we have a long way to go before we take it under control. Coming back to our topic today, founder of modern Turkish Republic, Mustafa Kemal Atatürk, said science is the most reliable guide for civilization, for life, for success in the world. And I think science is the only way for Turkey to learn the hard lessons from these events and move on, not to live the same disasters over and over again. And science is what we will be talking about today. I want to thank our FLICNA chair, Dr. Juhi Gupta from India. She works as assistant professor of pediatric neurology in SMS Medical College, Jaipur, India, and have been FLICNA chair since 2022 ICNA Congress. I will continue with introducing myself, our 2 speakers from Japan, and our guest expert, and leave the microphone for him. I'm Dr. Seçil Tunç, a last-year pediatric resident and an aspiring future pediatric neurologist, as I always like to introduce myself. My official training period here in pediatrics is completed. I currently work in Ankara Bilkent City Hospital as a senior pediatrics resident. In a couple of months, I'll be working in Boston Children's Hospital as a postdoc for a year in pediatric neurology, and then I will navigate my way through the field. I've been responsible from FLICNA social media handles since 2022 ICNA Congress as well. And I'm very excited and happy to be working with this group of amazing people, and also very excited to be participating today's talk with such a great expert and speakers. Our first speaker is Dr. Masashi Ogasawara from Japan. He graduated from medicine in Shinshu University in 2010, and then he worked as a pediatrician for 6 years in Hokkaido. Then he came to Tokyo to learn pediatric neurology and worked with Professor Nishino as a researcher at National Center of Neurology and Psychiatry, Tokyo, Japan, from 2016 to 2021. His work focuses mainly on researching clinical, genetic, and pathological findings on congenital myopathy, tubular aggregate myopathy, oculopharyngeal distal myopathy, and oculopharyngeal muscular dystrophy. Now he is working at the Department of Pediatrics, Showa General Hospital in Tokyo, and also at National Center of Neurology and Psychiatry as a researcher. He met our expert Dr. Schaaf at a congress earlier and helped us to invite him. Welcome, Oga. It's nice to have you. I think you're muted. Olga, I can't actually hear you.
Yeah.
No.
Hi everyone. Yes. Welcome to this session. I'm the speaker for this session, Masashi Ogasawara, from Showa General Hospital, Tokyo, Japan, and I'm very happy to organize these sessions. I'm looking forward to hearing from to talk from Professor Chris. Thank you very much for attending this session.
And our second speaker is also from Japan, Dr. Daisuke Sawada. In 2014, he graduated from Chiba University School of Medicine, Japan, and he has been a medical doctor for 9 years. He is a board-certified pediatrician in Japan. Since 2019, he has been a graduate student in Chiba University Graduate School of Medicine. His main research area is Werner syndrome, which, as many of our audience may already know, is a genetic premature aging disorder characterized by gray hair and hair loss, juvenile cataracts, diabetes, dyslipidemia, sarcopenia, and malignant tumors. He is also starting to study pediatric neurology and clinical genetics. He's interested in the process of human development. Welcome, Daisuke. It's great to have you.
Hello, everyone. Thank you for giving me this chance. So I'm very happy. And so I'm Daisuke Sawada from Chiba University Graduate School of Medicine in Japan. So I just starting to study pediatric neurology. So I'm not expert, but I hope to study this area. So they Professor Sharp, I'm looking forward to listen your good and great lectures. So and thank you for all.
Thank you. And our expert today is Professor Dr. Christian Schaaf. It's hard to introduce him in a short time, but I will try my best. Dr. Schaaf is the medical director and department chair of the Institute of Human Genetics at Heidelberg University, Germany. He is also a visiting professor at Baylor College of Medicine and the John Duncan Neurological Research Institute in Houston, Texas, United States. His laboratory at the University of Heidelberg is dedicated to investigating the genetic basis of neurodevelopmental and neuropsychiatric disorders. His focus is on translational research. All of his research projects relate to patients he has encountered in clinic. He is a solver of medical mysteries. By treating each patient with respect and curiosity, he has not only made a real difference in the affected individuals' lives, but also made seminal discoveries in the field of neuropsychiatric disease research. His work led to the discovery of multiple new disease genes, and 2 disorders have been named after him: Schaaf-Yang syndrome and Gauche-Bonstra-Schaaf optic atrophy syndrome, a neurodevelopmental disorder characterized by vision impairment and intellectual disability. His groundbreaking work has been recognized with many awards, including a Doris Duke Clinical Scientist Developmental Award, the William K. Bowles Award of Medical Genetics by Partners Healthcare and Harvard Medical School, the inaugural Selden Smith Award for Pioneering Research by the American Society for Clinical Investigation, and the Wilhelm Wieland Prize. He has authored several books, including Human Genetics from Molecules to Medicine, prefaced by a foreword written by Nobel laureate Dr. James Watson. He is a passionate teacher and educator, and a past chair of education for the American College of Medical Genetics. Most importantly, Dr. Schaaf is an advocate for his patients, affecting 10 individuals or 10 million. His work is aimed at understanding the cause of the disease, to empower affected individuals, and to improve the quality of life. He empowers families by helping them understand what human genetics has to offer in patient care, management, counseling, and family planning. Together with family support organizations, he works tirelessly towards care plans and therapeutic opportunities that will improve the quality of life, those affected with neuropsychiatric diseases. So these are the 2 syndromes that we just mentioned. And this is the article we read while Olga suggested us while inviting him for today's talk. He's a father of 4 and describes himself as a father, husband, physician, scientist, and writer on social media. Welcome, Dr. Schaaf. We are very excited to have you and very honored to have you. We are all at the presence of a rock star in the field at the moment.
Thank you. Thank you very much for the kind introduction, and also thank you very much for the invitation to speak to you all tonight. Greetings from Heidelberg, Germany. We just discussed about work-life balance. I just came back from home brought the kids to bed, and now I'm ready to talk about autism genetics. So let me share my screen. So I'm happy to talk about the association between autism spectrum disorders and genetic problems. First, autism spectrum disorder, as the name says, is really a group of disorders. It's quite heterogeneous, both clinically and genetically. Its main clinical characteristics are impaired social interaction, impairments in communication, repetitive and stereotyped behaviors, and restricted interests, and the symptoms have to be present in early childhood. It's very important to realize that autism spectrum disorder is and remains a clinical diagnosis. It's not a genetic diagnosis. So every once in a while, families come to our clinic in genetics and they say, we want to have a genetic test to find out if our child has autism. That is not the right approach. So only once autism has been diagnosed clinically, then it's our job in human genetics to help and find the underlying cause of autism. But there is no single genetic test that can prove whether the child is affected with autism or not. Autism has a high heritability, and we know this mostly from twin studies that were conducted in the 1970s and 1980s, showing that the concordance for autism spectrum disorder is much higher between monozygotic twins growing up in the same environment compared to fraternal twins growing up in the same environment. The concordance for autism spectrum disorders for monozygotic twins is anywhere between 85 and 95%, depending on the study, versus fraternal twins only show a concordance of 20 to 60% for autism spectrum disorders. Now, this can be used and entered into a formula to calculate the heritability of autism spectrum disorder. The FLICNA formula is one that we frequently use to calculate the heritability of traits or disorders. And these studies suggest that the heritability of autism spectrum disorder is between 60 and 90%, which at the same time means that the environmental impact is estimated to be 10 to 40%. Now, how is this possible? Sometimes families ask me, why do you say that there is a high heritability of autism spectrum disorder? When we look at our family, it doesn't look like it's inherited at all, because the child that we're bringing to your attention is the first one to be affected within our family. So it's important to explain to them the concept that a genetic disorder does not necessarily need to be inherited and passed on from one generation to the next, but there is still the possibility that the heritability is high. Now, why is this? Because of multiple effects. First, many cases of autism spectrum disorder for which we identify the underlying cause are due to de novo autosomal dominant mutations. So the affected child is the first one in the family because it's a new mutation that occurred either when the sperm or the egg cell was formed. Second, there are occasions of autosomal recessive inheritance, where the trait is passed on from both mom and dad, and only in the homozygous or compound heterozygous state, the child will be affected. Occasionally, we also have X-linked inheritance, which can be passed on from one generation to the next as long as it is passed on in the female line, and only when a mother passes on the mutation to her son, and in the son, the mutation is hemizygous because of his single X chromosome, he will be affected. And then ultimately, there are also cases of oligogenic or polygenic inheritance, meaning that subtle mutations or genetic variants are passed on from both mom and dad, and when they accumulate to pass a certain threshold, then the child may manifest autism spectrum disorder clinically. So what I'm trying to show, ASD is a group of disorders. The etiology of ASD is mostly genetic, but autism spectrum disorders are rarely inherited. Another reason why they're rarely inherited is the fact that due to their social interaction impairment, individuals with autism spectrum disorder are less likely to have children compared to neurotypical individuals. And it's not just clinically heterogeneous, but the genetic basis of ASD is also complex. We've learned this the hard way over the past 10, 15 years, as hundreds of autism risk genes have been identified. In fact, as of today, more than 1,000 genes have been identified, which are considered to potentially be involved in the etiology of autism spectrum disorder. And I'll go more into this. At the same time, there is no single gene that accounts for a large proportion of cases, because obviously there could be a scenario where one gene accounts for 30–40% of all cases, and then all other situations are rare. But that does not seem to be the case for autism spectrum disorder. Each and every gene rarely accounts for autism spectrum disorder. There has not been a single gene that has been identified to date that would account for more than 1% of all cases of autism spectrum disorder. And the autism genes, they are scattered all over the human genome. There is no particular hotspot or chromosome where most of those would be located. There is a substantial number of autism genes on the X chromosome, in part because they were relatively easy to study historically. And, we may have time to go into this a little more. There is a male predominance in autism. Boys are affected approximately 4 times as frequently as females, and the autism genes on the X chromosome are one, but only one reason why this is the case. There are additional reasons, and we may have the time to go into those a little later. So there are some people in the field that say autism is a snowflake disorder, because when you look from a macroscopic level clinically, a lot of kids with autism spectrum disorder, they share a lot of features, and they somewhat look alike. But when you look with high resolution genetically, they almost all of them are distinct and unique in their own way. Just like snowflakes all, make snow, and it's all crystals of frozen water making snow. But when you look at them under the microscope, the structure of the snowflake is individual. There are several intriguing features of autism genetics that can generally be used for neuropsychiatric genetics. And I want to highlight them because these are terms and concepts that are important to understand neuropsychiatric genetics at large. So I'm just giving you an example of a pedigree of a family with manifestations of psychiatric disease. And I'm showing the clinical indications written next to the respective individuals. And I've highlighted individuals who have been identified to carry a mutation in gene A in light gray. And then there's also an individual in the pedigree that has another mutation in a gene B that is highlighted in light blue. So let's take these 2 individuals highlighted with a red arrow. They carry the same genetic mutation in gene A, yet they manifest different psychiatric diseases. The son has been diagnosed with autism, and his mother has been diagnosed with bipolar disorder. We call this phenomenon pleiotropy. Pleiotropy means that mutations in the same gene can be responsible for the development of different disorders. And in fact, we see that in autism genetics a lot. In particular, we see a lot of overlap between the genetics of autism spectrum disorder and schizophrenia. So a lot of autism genes have later also been identified to substantially increase the risk for schizophrenia, even to the point that Kathy Lord, who's one of the most famous psychologists for autism, wrote an editorial once where she asked whether schizophrenia is on the autism spectrum. Then we have another phenomenon highlighted in this individual here, this female person. She carries the genetic mutation that provides a risk for psychiatric disease like autism and bipolar disorder, yet she has been phenotyped clinically and she does not manifest any of those disorders. We call this incomplete penetrance, and we see this a lot in psychiatric genetics. Incomplete penetrance means not every individual carrying the genetic variant that predisposes to a clinical phenotype necessarily manifests the phenotype. And then we see these two individuals, mother and daughter. They both affected with bipolar disorder, but the clinical severity is very different. So in the mother who already passed away, she had bipolar disorder. She was hospitalized 4 times throughout her life, and she committed suicide at the age of 42. Her daughter also manifests bipolar disorder, but it's considered relatively mild. She's never been hospitalized, and it's fairly responsive to medications. So they both penetrant. They both manifest the same disease, yet the clinical expressivity is very different. So we call this variable expressivity. And this means that the phenotypic severity varies, even though the underlying genetic predisposition is the same. Let's go more into the genetic heterogeneity and complexity of psychiatric disease, and in particular autism spectrum disorder. So we have to realize that hundreds to thousands of genes are involved in the etiology of autism. There are gene-gene interactions, and to make it more complicated, there are also gene-environment interactions. When the genetic impact is severe enough, even with a very supporting environment, the genetic variant may be strong enough to lead to the manifestations of autism spectrum disorder all by itself. But we also know that if the environmental impact is strong enough, without any genetic susceptibility, that can also cause autism. So in primates, studies have been done where baby chimpanzees have been separated from their mothers and isolated right after birth. And if you let them grow up in social isolation, most of those monkeys will manifest the features of autism spectrum disorder. We also saw this after the Iron Curtain fell in the 1980s and 1990s in orphanages in Romania, for example, where little orphans grew up in the orphanages in like severe social deprivation and isolation with very little to no social attention that was given to them. And the prevalence of autism spectrum disorder among those young children in those orphanages was up to 40%. So you can see that when the environmental impact is strong enough, you don't necessarily need the genetic predisposition to manifest the features of autism spectrum disorder. Now, the children who come to our clinic, most of them, by and large, have grown up in a very supportive, socially interactive environment. and yet they manifest autism spectrum disorder and oftentimes other neurological disorders as well. And it's our task in human genetics to help those families find the underlying cause of their children's disease. Now, some of the questions that I want to answer in the next few slides is, how do we decide which genetic testing is appropriate for those children and which genes should be assessed as part of the genetic testing? So one clinical challenge is that the indication autism is used excessively, and it encompasses a broad range of clinical manifestations. In some countries, I would say in particular in the United States, I worked in the United States for 12 years, families also seek the diagnosis of autism as it opens the door to additional therapies and services. And I cannot emphasize enough that autism always remains a clinical diagnosis and not a genetic one. The genetic testing landscape for autism has changed dramatically over the past 20 years. 20, 30 years ago, karyotype analysis was almost the only test that we had available in the genetics clinic. And with karyotype analysis alone, we will only identify the underlying cause for autism spectrum disorder in 1 or 2% of cases. Then Fragile X syndrome was identified, and autism spectrum disorder is a classic manifestation of Fragile X. For a long time, Fragile X was recommended on the guidelines for molecular testing for autism spectrum disorder. It probably should not, and I can go more into this, as Fragile X only accounts for a small subset of cases, and typically those families do have a positive family history, either of Fragile X syndrome, autism, intellectual disability, or at least of Fragile X-associated tremor ataxia syndrome or premature ovarian failure. The first real game changer in the autism clinics came in the late 1990s when array CGH, and especially in the early 2000s when array CGH was introduced. With chromosome microarray analysis, we can identify the underlying genetic cause in 10 to 15% of all individuals with autism spectrum disorder. Then we had next generation sequencing come about, and we have the choice between multi-gene panels and exome sequencing. And using both or one of the two, we can increase our diagnostic rate to a total of approximately 50%. So in clinical practice, and I will talk more about this, we now typically use array CGH and exome sequencing, and combining those two, we get to a diagnostic rate for all children with autism spectrum disorder who come to our clinic in approximately 40 to sometimes 50% of cases. What do the guidelines say? The guidelines, this is one of the intrinsic challenges of guidelines. They usually way too old, and the methodology in genetics develops much faster than the guidelines can be written. So the practice guideline by the American Academy of Neurology and Child Neurology says, in affected individuals with autism spectrum disorder, you should do karyotype analysis, Fragile X testing, and metabolic screening. Now, this is so outdated, I actually recommend not to do any of those 3 tests in the average child with autism spectrum disorder, only if you have specific indications that I can go into a little later. The American College of Medical Genetics guideline from 2013 says, in every individual with autism spectrum disorder, you should do chromosome microarray analysis. In males, you should do Fragile X testing, and you should consider metabolic screening if you have any diagnostic clues that it could be metabolic. For example, when you see developmental regression in those children. And then second-tier testing could be specific gene testing like MECP2 sequencing for Rett syndrome or PTEN sequencing, especially if you have macrocephaly in your affected individual. The American Academy of Pediatrics has the currently most up-to-date guideline for autism spectrum disorder, and they recommend chromosome microarray analysis, consider Fragile X in males, consider MECP2 in girls, consider referral to genetics, and the workup might include whole exome sequencing. And this is close to reality nowadays. I think in most of the first world countries, for patients with autism spectrum disorder, we will do chromosome microarray analysis, oftentimes as a first tier test. And if that's negative, we do exome sequencing. Because the diagnostic rate of exome sequencing is higher than the diagnostic rate of chromosome microarray analysis, we may also flip the order, and that's actually what we do in my practice now. We start with whole exome sequencing, and if that's negative, we resort to chromosome microarray analysis. Now, I want to introduce the methodology just a little bit. Chromosome microarray analysis, there are two different technologies that we use. One is based on single nucleotide polymorphisms, and the other one is called Array CGH, which stands for Array Comparative Genomic Hybridization. And it's called comparative because the amount of genomic material is compared between a patient sample and a control sample. Both genomic DNAs are fragmentized, and then they are labeled with fluorescent dyes. They are mixed and they are hybridized to the actual array that has the genomic sequence of hundreds of thousands of specific regions of the human genome. And these two samples with the fluorescent labels will compete in binding in the respective wells. And when you wash away whatever's left, only the sequence that binds to the well will be left. And then the fluorescent signal is measured, and you can compare how much fluorescent signal you get from the patient sample and how much you get from the control sample. And the computer can read all those hundreds of thousands of samples, and it will give you an array plot like the one that you see here on the right side. And it will show any deviations from normal, either lost material as a deletion or gains of chromosome material as duplications or triplications. And this test is so powerful because its resolution is much higher than anything that you were able to get with the classic chromosome analyses. So karyotype analysis has a resolution of 4 to 5 megabases, meaning that the chromosomal aberration has to be at least 4 to 5 megabases in size so that you can see it under the light microscope. Chromosome microarray analysis has a resolution of approximately 5 kilobases, which is 1,000-fold higher. And this is how I compare this to my patients. Chromosome analysis is like looking at a map of the world. And obviously, there's a lot of information that you can get. You can see Australia is present. You can see Iceland is only present once and not twice. But the resolution is quite limited. So, when you want to look at a street or a town, the map of the world doesn't really help you. And nowadays we use Google Maps. So technology is helping us getting a higher resolution. And this is what the chromosome microarray analysis does. So here on the right side, you see Google Maps with 1,000-fold higher resolution compared to the map of the world. And now suddenly, we can see Heidelberg, where I'm located and where I'm speaking to you at this time. So the other test that's really important because it's widely used and continues to be more widely used across the world is exome sequencing. The technology is very different. The information that you get is very different. So here we look at single nucleotide changes. It's called exome sequencing because it's enriched for the sequences of the coding genome. Only the exons of the protein coding genes are looked at. This is approximately 21,000 genes, and it only looks at approximately 1% of the entire genome. We need genomic DNA for this test that can be gathered from any sample that contains cells that have a nucleus. So most frequently blood samples from which we use the leukocytes, but we can also use saliva, we can use fibroblasts or any other tissue that contains genomic DNA. The information between chromosome microarray analysis and exome sequencing is very different. So think of your genome as a library. The chromosome microarray analysis only looks at the substance and only looks at the material that's there. So it's like doing an inventory of the library, counting the number of books and checking if each copy of the various books is present in the right amount that is supposed to be there. Chromosome microarray analysis does not look at sequence at all. This is what exome sequencing or any other sequencing technology does. It opens the books and checks for typographical errors, single letters that are changed in the genetic code or the text of the book. I mentioned already that autism spectrum disorder is genetically very heterogeneous. As of now, there are more than 1,000 genes listed in the Safari gene database. The Safari gene database, which is curated by the Simons Foundation for Autism Research, is one of the most widely used resources to look at autism genes. And it categorizes the autism risk genes into different evidence levels: high, strong, suggestive, and then lower levels of evidence. And you can see that more than 900 genes are in the suggestive, strong, or high range for autism evidence. But this is only one database, and there are many different resources that you can use. And one of the real challenges that we face in autism genetics is that there's little agreement and little overlap between the different databases and the different resources. So one paper that Cecil mentioned before is the paper that we published in Nature Reviews Genetics in 2020. At that time, we looked at high quality resources for autism genes. And we found that there's limited overlap in the bona fide autism genes between those different resources. Also, I serve as a co-director for the Autism and Intellectual Disease panel for the ClinGen initiative. And for ClinGen in 2019, we looked at the genetic testing registry for all gene panels for intellectual disability and or autism spectrum disorder. At the time, we identified 65 panels and a total of almost 5,000 unique genes that were on those testing panels. And after very strictly removing all panels that were either too broad in scope or focused on non-neuronal indications, we were still left with 30 panels and 971 unique genes. However, out of those 972 genes, 51% of all genes appeared on one panel only, really highlighting the fact that when you as a clinician order an autism next generation sequencing panel for your patient, the information that you get may be very different if you order in lab A or lab B, or whether you send your panel to lab C. There's little overlap between. There are certainly some genes that appear on almost all of those panels, but then the specifics of the panels may be very, very different. Now, this highlights the need to carefully curate gene-disease relationships and to have experts evaluate the evidence that exists for the association between genes and the respective disorders. As part of that, I mentioned I'm one of the co-directors of the ID Autism Working Group for ClinGen. ClinGen is an NIH-funded resource that defines the clinical relevance of genes and variants for use in precision medicine and research. And up until today, more than 2,000 genes have been curated. In our working group, we have 28 members, and more than 100 genes have been curated for their specific relationship between ID and or autism. And I want to present you the workflow of how we think about the curation of autism genes in the ClinGen SOP. So we are a group of experts who are interested in the genetics of autism spectrum disorder. And first we pre-curate. So we look at a gene and we decide which clinical entity we want to curate under. So we need to define the gene and the respective genetic entity. Then we perform a very in-depth literature search trying to pull all information published to date about the relationship between that gene and the respective clinical phenotype. And we curate two different kinds of evidence. First, genetic evidence, and second, experimental evidence from animal models, cellular models, or others. Then all of this is pulled together, assessed, and scored in the ClinGen framework. And then we discuss the curation in our group of experts. We then assign a validity classification, do the expert review, and then after the expert review, we approve the curation and we publish the final classification on the ClinGen website. We have different evidence levels. It's a very time-consuming process, but we feel that it's important because it's the most thorough process to date to decide on gene-disease relationships. The different evidence levels are disputed or refuted, limited evidence, moderate evidence, strong evidence, and when strong evidence is provided, and there's also replication over time, then strong becomes definitive evidence. Now, I mentioned the top autism genes from multi-gene panels, and we actually went through that exercise, pulling those autism genes that most frequently appear on next-generation sequencing panels, and we find a definitive evidence for approximately 75% of those top autism genes. But you also see that there are genes with only moderate, limited, or even disputed evidence among the top autism genes from multi-gene panels, suggesting that there are problems within the system such that when a gene is proposed as an autism gene, it may be replicated over and over again in the medical literature without actual evidence, and many labs may consider this a bona fide autism gene without going back to the evidence and really look at, how strong that evidence is. Another point that I want to make is that rarely do we see genes that only cause autism spectrum disorder. So here are the various disease entities that we've curated under in the ID Autism group. And you see that we curate for a lot of specific syndromes, but the less specific entities, are rarely autism alone, not that often intellectual disability alone, but most frequently are actually complex neurodevelopmental disorders, meaning that autism often co-occurs with other comorbidities, such as intellectual disability, epilepsy, ADHD, or others. And this has also been shown in a publication by Myers et al. In the American Journal of Human Genetics that I want to highlight. They looked at many different autism genes, and they show that, autism only is only seen in small subsets of cases. ID only is seen in subsets of cases. ID and autism together are seen in subsets of cases. But when you look across all the genes, you will not find a single gene that has ever been reported in ASD alone. All genes can manifest with autism more heavily, with intellectual disability more heavily, but there is no gene that really only causes autism by itself. We think that there's a fine balance of neuronal function, and that the human brain can manifest clinical features only in very few ways, like cognitive impairment, social impairment, anxiety, etc. So there are limited ways of manifesting brain dysfunction. And we believe that this limited dynamic range is one of the reasons why we see variable expressivity and clinical gradients for most neurodevelopmental disorder genes. It's also important to realize that more does not necessarily equal better, and that deletion and duplication of the same chromosomal region can manifest neuronal dysfunction in very similar ways. So we see for many chromosomal locations that both the deletion of that region and the duplication of that region both increase your chances of developing intellectual disability in ASD. While, a priori, you might have thought when the deletion causes cognitive impairment with intellectual disability, then the duplication could potentially cause increased cognitive function, but that is very rarely the case. We think of it as the Goldilocks principle of neuronal homeostasis. So some of you may know this American book or fairy tale, Goldilocks and the Three Bears, where Goldilocks comes to the house of the three bears and she finds that house empty. And to make a long story short, she finds food to eat, and, the oatmeal that she finds, one of the bowls is too cold, she doesn't like it. The other one is too hot, she doesn't like it. And only the third is just right. And then she tries to sleep in the beds of the bears. One is too hard, one is too soft, and one is just right. So the Goldilocks principle means that either extreme is not good, but there is a sweet spot that is just right. And we feel that this is true for a lot of neuronal function in the human brain. Too much isn't good, too little isn't good. It needs to be just right. Now, with all that I've said, it sounds very complicated, and we may ask the questions, do autism genes even exist? What is the level of evidence if we curate for autism only? Because I showed you that very few genes manifest autism only. So this is part of the work that we are highlighting in this Nature Reviews Genetics article that I wanted to show you. This is the work of an international group of experts in the field of autism. And we came together in Toronto in 2019, trying to find the best way to curate autism genes. And many different approaches were presented. And it was felt that the ClinGen approach was closest to the perfect approach of curating autism genes, but it was still not perfect because it was taking genetic evidence into consideration. It was taking experimental evidence into consideration, but it did not focus very specifically on the value of clinical information. And for autism, there are wide ranges of how deep that clinical information can be. Some people publish and they say, oh, there were autistic features in that patient, but what does that mean? Others say, well, the DSM-5 criteria for autism spectrum disorder were met. That's worth more than just autistic traits, but it's still not the gold standard of diagnosis. The gold standard of diagnosis would be an ADOS, Autism Diagnostic Observation Scale 2, and the ADIR, the Autism Diagnostic Interview Revise. And when ADOS and ADIR are both meeting the threshold for autism, then yes, you do have the gold standard diagnosis of autism. So we felt that this clinical gradient and the richness of the clinical information should also be taken into consideration. And Jacob Forstmann, who is a child psychiatrist from Sick Children in Toronto, devised the criteria of how this clinical information should be taken into consideration in the ClinGen framework. And we came up with the so-called EAGLE score, which is probably the most stringent way to curate autism genes. And for those genes for which the EAGLE score has been calculated, you can see that in the Safari Gene website of the Simons Foundation for Autism Research Initiative now as well. So when we curated 80 genes from the Safari category 1, which is the highest level within the Safari Gene consortium, we found that approximately 30% of them had the richness of information to get to definitive evidence in our EAGLE framework. Approximately 10 to 15% were in the moderate range, 25% in the limited range, and a small subset even ended up in the disputed range. So this really highlighting that if you are very strict and if you want the highest level of richness and evidence, then the number of bona fide autism genes decreases. But if you're interested in this, feel free to read our article in Nature Reviews Genetics and also look for the EAGLE score on the Safari Gene website. Now, on the other hand, this is also important information when you are publishing genetic findings in individuals with autism, because if you do not include the information that we as curators are looking for, those cases may not get the highest number of points because we're missing information. So please, when you publish genetic findings in individuals with autism, include the genetic methodology that was used. Was it a targeted gene panel? Was it a targeted gene approach with only one gene sequence? Was it exome or genome sequence? Please include information whether you know if the mutation is inherited or de novo, because de novo cases that were subjected to exome or genome sequencing will get much higher scores than inherited cases, especially if they inherited from unaffected individuals. And also include the clinical information, which assessment was used to establish the clinical diagnosis of autism spectrum disorder. Now, why even bother? Why do we spend so much time about curating autism genes? First, we feel it's important in order to decide which gene panel to use. And if you're using whole exome sequencing, in many cases, virtual gene panels are used based on whole exome sequencing panels, and I will show you and explain that in a minute. It's also important to have bona fide autism genes in order to give anticipatory guidance following molecular diagnosis to the affected individuals and families. It's important to get into a position where you can inform yourself and the families from genotype to phenotype and to interpret variants of uncertain significance. Now, I briefly mentioned virtual panels. This is an approach that is being applied more and more widely. We're getting away from NGS gene panels with limited number of genes, and instead, many laboratories now just sequence the entire exome, but then they use bioinformatic filters, which we call virtual gene panels, only filtering for variants that reflect the clinical phenotype. So, for example, if our patient has autism spectrum disorder and macrocephaly, we will filter within the exome sequencing data only for variants that have been described in the context of macrocephaly and autism spectrum disorder. And ideally, the lowest hanging fruit will be those variants in genes that cause both macrocephaly and autism spectrum disorder. There are more advantages of exome sequencing other than just being comprehensive and also having the flexibility of virtual panels. They also allow for reanalysis, and this is really meaningful because continuously every year, dozens and dozens of new genes, new disease genes, new autism genes are being discovered. So even if your exome sequencing approach is negative today, that autism gene in your patient may be identified next year. So if you reanalyze the exome sequencing data in 2 years from now, you may find the underlying cause of your patient's autism while you're not able to identify it today. And the exome sequencing data are typically stored for many years. And without doing the test again, you can just reapply the bioinformatic filters in 2 years and do the reanalysis, and you may be successful in identifying the underlying cause. So in summary, I tried to show you that autism spectrum disorder is genetically very heterogeneous, that gene disease evidence needs to be carefully curated. I tried to show you that chromosome microanalysis and exome sequencing are the genetic tests of choice for individuals with autism. That I prefer exome sequencing over gene panels, and that you should consider incomplete penetrance and variable expressivity and other of those genetic phenomena when you meet your patients and try to interpret their genetic information. With this, I would like to thank the members of my lab, but also my collaborators, especially the people from the ClinGen group that worked tirelessly on the curation of those gene disease associations. And with that, I'll finish, and I'd be happy to take any questions that you have. Thank you.
Thank you for the fantastic talk. It was really great to listen to you. And I feel that we should actually study a little harder on the topic and invite you once again so, to learn more about in detail maybe. I see no questions on the chat box, but I invite any of our attendees, if they have any question to ask, they may ask right now. And I want to ask if Oga and Daisuke have any questions.
Thank you for wonderful talk. I learned a lot from you. And I have one question. So you said that if you perform microarray and exome sequencing, and you will get the maybe possible mutation in 40 to 50% of patients. Is there any difference between the, so far I saw that if the patients have ASD plus and for example leukodystrophy or severe epilepsy or polymicrogyria, maybe you can get. I thought that we can get mutation in high frequency, and if the patient has only ASD and without any severe severe brain abnormalities or something, there might be the low. I think if we perform exome sequence of a microarray for these patients, maybe we will get the results in lower frequencies. It is right, or what do you think?
Yes, that's absolutely right, Olga. The diagnostic rate is higher in cases of complex autism spectrum disorder. So we do know that the lower functioning the patient is, the higher the diagnostic rate, meaning that in individuals with high functioning autism or previously considered Asperger's syndrome, the diagnostic rate with these tests is much lower than in individuals with intellectual disability and autism. And if you have additional features like congenital malformations or specific findings on brain MRI and or epilepsy, then your diagnostic rate goes, way up. So you can use that in a clinical setting when you have enough experience. Oftentimes the families want to know what's the chances of finding the underlying cause. When you have a leukodystrophy, for example, the likelihood of finding the underlying cause may be as high as 60, 70%. On the other hand, in a high-functioning individual with autism spectrum disorder and an IQ of 120, the diagnostic rate may be as low as 15%, even with the two tests combined. In fact, we did a study several years ago to look into the genetic causes of high-functioning autism. And we believe that the same genes actually play a role as in low-functioning autism, but hypomorphic genetic variants in those genes appear to play a role. And there seems to be a model of oligogenic inheritance, maybe with a sex-specific threshold. It's the so-called female protective model. We know that the female brain can tolerate more genetic burden than the male brain prior to manifesting clinical autism spectrum disorder. And there's also a Nature paper that just came out maybe 2 weeks ago, looking into the effects and consequences of rare variants in relationship to common traits. And it kind of shows the same concept that the same genes seem to play a role, but it's different types of mutations.
Thank you. I see one question from the chat box. What should we consider if there is no dysmorphism present, CMA or WES? If there is no clue at all, what should we consider first?
Yes. So, you know, I think the vast majority of patients that we see in clinic with autism spectrum disorder does not have very unique dysmorphic features. So it's relatively rare that they have a clinical facial manifestation or clinical course that is so typical that we are brave enough to order just a single gene. You know, these are situations like, you know, you have autism and macrocephaly with +3 standard deviations and freckling on the glans penis that you think of Benny and Riley Ruvalcaba syndrome and you order only P10 sequencing. Or you have a post-pubertal individual with low functioning autism spectrum disorder and macroorchidism and you order only Fragile X syndrome. Or you have a girl with secondary microcephaly and developmental regression and washing hand movements, and you order only MECP2 sequencing. The vast majority in my clinic, I do not have a clue when I see them what the genetic cause would be, and we go with the genome-wide tests. It depends on reimbursement. You know, up until 3 years ago, here in Germany, we would have to order chromosome microarray analysis first, and then exome sequencing only in select cases. Now at university hospitals, we are allowed to start with exome sequencing. And because it's the test with a higher diagnostic rate, a priori, I actually now start with exome sequencing. And then if that's negative, I go to the array. But that also only because exome sequencing is not perfect at copy number calling. Next year, here at the university, we are switching completely from exome to genome sequencing. And genome sequencing has much better copy number calling. So in that case, we can do it all in one test. We don't need to do exome and array. We just do genome sequencing and we get information on both copy number and also single nucleotides.
We actually have another question from Hiroya Nishida asking, would you recommend all autism spectrum disorder patients to have genetic tests if it's possible?
So this is a hard question. You know, I certainly recommend genetic testing for all individuals who have low functioning autism. And these are also the individuals who are typically referred to our clinic. We get a smaller number of individuals with high functioning autism. And for those with high functioning autism, it's more challenging to make those recommendations. So in those situations, I try to spend more time on counseling the families and also discuss with them, you know, which benefits they expect from a molecular diagnosis. I also talk about the challenges of genetic testing, meaning, you know, the challenges of getting a variant of uncertain significance. You know, what does that mean? What would they do with it? You know, sometimes getting a variant of uncertain significance increases the uncertainty because then, you know, the family is left with, you know, I don't know what this is. It makes me concerned, but I can't do anything about it. But, you know, there are clearly some families who feel very guilty about having a child with autism. They wonder and they blame themselves that they may have caused it. Because I don't know, like, you know, everybody comes up with their individual explanation because they didn't just eat organic food during the pregnancy or because they had a glass of wine before they knew they were pregnant and they blame themselves. Is it related to the wine? I always try to take the guilt away from them and say, you know, these situations typically are not a justification, you know, for the autism in their child. But these may be situations where we can help with providing a molecular diagnosis. But it involves a lot of counseling and discussion between physician and patients as well.
I see one question. Paula Ivarola asked that they would like to know if you study patients with tuberous sclerosis because they have a really wide variant of clinical expression.
Yes, that's absolutely true. So individuals with tuberous sclerosis clearly manifest variable expressivity, sometimes even manifest incomplete penetrance. But you know, the classical manifestations with the skin findings and the tubers on brain imaging, plus you know, epilepsy and autism spectrum disorder, very rarely do you see a patient who manifests all of those features. And sometimes, you know, we've been surprised because the child is brought to our attention with fairly typical manifestations of TSC. And looking at the parents, you know, we would feel that clearly they're not clinically affected, but then we find the variant in one of the parents as well. And then, you know, when we look more carefully, for example, with the Woods lamp, we find the white spots in the parent as well, but, you know, cognitively and neurologically, they're not affected at all.
And I see our chair, Dr. Juhi Gupta, has a question. I would like her to ask her question.
Thank you. Thank you, Dr. Shah, for such an amazing presentation. We were enlightened by your talk. So in my clinic, I have parents of children with autism visiting, asking me if the further children are likely to be affected. So they usually request to do a genetic test in their child and let them know what are the chances if they conceive. And some of them are already into first trimester and they want to know if the baby can be affected. So what do you tell these parents? And if they're already into pregnancy, do you send the whole exome and CMA together?
Yes. So this is very challenging, and I don't think there's an easy answer to this, right? When I see patients in my regular genetics clinic, this is something that we talk about a lot, is future family planning. And it's always better to do genetic testing on affected individuals in a controlled setting where there's no time pressure, like during the pregnancy. You know, in the pregnancy, you know, when they are already pregnant and they have an affected child, we have those situations occasionally, and we send rapid exome sequencing and or microarray on the affected child when it's a severe neurological disorder manifesting with autism spectrum plus. We typically don't do this for high-functioning autism in those kind of time-sensitive situations due to the given concerns and the chances of variants of uncertain significance, which then would be troublesome during the pregnancy as well. So here in Germany, we have like a legal situation where molecular testing is also regulated by the gene diagnostics law, and that gives us also some guideline about, you know, genetic testing prenatally and on affected family members. We would only offer prenatal testing for pathogenic or likely pathogenic variants with a clear gene-disease relationship. We would never offer prenatal genetic testing or pre-implantation genetic testing for that sake on variants of uncertain significance.
Right. Thank you for your answer.
I don't know if Oga and Daisuke has any other questions, because I interrupted with other questions. Do you guys?
I have questions. So in chat, so another question. So we have
Yeah, I saw it, but if you have a question
I
you want
see.
to ask, you can go ahead and we can ask it later. If
I see. So, thank you for a great talk. So, um, I feel that the curation process is very difficult and challenging work. So, especially to conclude that L'Italia's variant account for phenotypic finding of ASD. So, I learned that you propose a systematic evaluation framework model for making list of genes relevant to ASD. So I believe that since only genes with higher evidence are selected for the list, the framework model is considered to be very useful for clinician. My question is, so at present, how far along is the creation of that framework model? So how progressing and improved? And so in addition, how is the validity of the framework model?
Yes. Very good questions. Because it's such a time-consuming process, and because there's such a large number of potential autism genes, we're not moving at the speed that we would like to. But approximately 200 autism genes have been curated to date. And every newly curated gene is then also presented on the ClinGen website. We started as one curation group, and we meet for our expert discussions every other week, meaning twice a month. Because we felt that we were moving too slow, and we were able to attract more expert members to our group, we then split into 2 groups, and then most recently actually split into 3 groups. So now we are able to proceed with 3 times the speed. On the other hand, the curations also become more difficult because we started with the top autism genes that are present on the largest number of autism panels. So with that, initially, we curated a lot of genes that were strong or definitive evidence. Now that we're moving lower in the rank of those gene panels, we are getting more and more genes in the moderate or limited range, which require more discussion within the group, really looking very carefully at the evidence that exists. But we still feel that this is the right thing to do because we feel it's helpful to the clinicians and the families. And what's important to realize is once you've finished whatever that number is, you're not done with the work because you have to start all over again, because in the meantime, additional evidence has emerged. So we now actually have, we already have the first handful of genes that we are recurating because we realize that additional important information has been published that needs to be taken into consideration. So it will be interesting to see at this point, we feel that we are still superior to all kind of bioinformatic algorithms that could potentially be used. But with artificial intelligence and neuronal network learning, we may soon be in a situation where the AI may be just as good as we, and then we can probably stop or just kind of reassess the evidence that has been curated by AI.
So I'm very impressed with your research. Thank you.
Thank you.
We have one last question on the chat box, if you still have time. Denise Garlaki is asking, how is the percentage of autism spectrum disorder gene has been discovered right now among all the autism spectrum disorder patients?
So I'm not sure if I understand the question correctly. I would say among all autism patients, it heavily depends on your cohort. I think the diagnostic rate in our clinical setting in human genetics, we do identify the underlying genetic cause in approximately 40 to 50% of cases, as I mentioned, but that has some ascertainment bias because we see more low-functioning autism individuals. Among all individuals, I cannot tell. I would expect that, you know, I think for all autism spectrum disorder diagnoses, approximately 60 to 70% of individuals who are diagnosed are considered low functioning, and 30 to 40% are considered high functioning. But I believe that in the population, there's a lot of high functioning individuals with autistic traits who do not seek clinical diagnosis because they don't suffer as much. So I think, you know, my estimations are based on clinical cohorts rather than population-based cohorts.
Yeah, thank you. And if there is no other question from Juhi, Oga, or Daisuke, I think we can close today's session. Thank you for your time joining and enlightening us about